So welcome to this lecture. I guess you guys know this is P2 by now. Josh asked me to talk about biomedical big data and data science. So this lecture is an overview. Hopefully you'll find it interesting and or useful. If not, please tell me so I think I'm going to do it next year. So it's make it better. You'll also see me later in the semester talking about something else. Probably graphs and brains. Okay, so let's just get started. Here's biomedical, big data, and data science. Feel free to ask questions, interrupt, tell me I'm wrong, or dumb, or whatever. Just like feedback and interaction. So, I made this, can you guys read this? Okay, I made this slide. It's a brief history of data science. I don't know if anybody, do you guys recognize these terms? I don't really know where you are in terms of your math, stats, data science realm, but 0? Okay, so does anyone recognize any of these terms? Just yell out enough to recognize. Numbers anything else? Anything other than the numbers? Stones? I think it's a different stone. Besides names or numbers, any concepts? Bayesian stats. Okay. So, all right. Well, I would recommend you learn all this stuff. I'm not going to teach you now, but you come to my class, you'll learn a bunch of it. Is that true? Okay, so I thought I'd start defining some terms that people throw around a lot in the last few decades that are closely interrelated, just to give you a sense of what everyone's talking about. So statistics, I literally just took the first sentence from Wikipedia for all of these definitions. So this is not like from God or whatever. From Wikipedia. Do with that as you wish. I think actually it's pretty good definition. Statistics is a branch of mathematics dealing with data collection, organization, analysis, implementation, and communication. Statistics basically started a few hundred years ago. It's called statistics because it was funded by the state. The government wanted people to do some stuff like census stuff. It's a bit antiquated of a name, but it is. Data science is a more recent term, interdisciplinary field, where scientific methods, algorithms, blah, 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 to extract knowledge and insights. That's great. Pattern recognition is another one. Data mining is another one. Machine learning is another one. People really like saying data science and machine learning these days. They used to like saying pattern recognition and statistics more. They're all more or less the same thing. They have slightly different foci. Different people use the terms, partially because they kind of emphasize one thing more than the other. For the most part, they're interchangeable. But anyway, those are the things that people mean when they say data science. So there's a new field called biomedical data science. And the reason you know it's new is because there's no real departments in academia that are called that yet, although they're starting to be. So Stanford started a new program, and they define, again, like the first sentence of their website, the study of biomedical data and information, how such data and information may be structured, how analysis process, blah, 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 whatever, healthcare. Okay? University of Wisconsin-Madison, they have another field, another degree for biomedical data science. It's an interdisciplinary field that encompasses all that crap to improve healthcare. I didn't like those definitions personally, so I made up a new one. What I did was I took all the parts I liked from all of these things, which are more or less equivalent, and then parts I liked from these things, which are more or less equivalent, and made up a new one that hopefully, as we go through the rest of the slide, you'll see it's a meaningful and coherent definition. So it's an interdisciplinary field that uses algorithms, statistical models, and database systems to manage, visualize, wrangle, summarize, journalize, and control biomedical data, driven by efforts to improve health and healthcare. And the reason I use that is because these things are the things I'm going to emphasize, is things that you do in biomedical data science, things that one needs to have tools to do and kind of insight in order to do. Okay, so this is my operational definition. Does it make sense to you guys? Does anybody want to fight with me about what you think biomedical data science is? there are like actively lots of fights in BME even about what we mean. Because we just started this new focus area, it's a PhD focus area, and an undergrad one, a master's program. And different people have different beliefs, and they're all fine because you can make up whatever definition you want. If anyone thinks I'm missing something or overstated something, now's a good time. Okay. So why is biomedical data science hard? So there's this other term I didn't bother defining called big data, which comes up a lot. And people talk about big data. There's 4 V's of big data. So volume basically is a bunch of data. In terms of like electronic health records or in databases, they're called records. You can have millions and billions of records. Variety. Often big data, there's lots of different kinds of data. So again, in biomedical context, there could be, you're a patient, and so you've got all these MRI scans and then some CT scans, and then maybe you answered some questions on when you filled out basic patient history forms, and then there's some blood tests, and then maybe they sequence some of your DNA, and that's all different kinds of data that are all associated with the same person. Velocity, again, in biomedical healthcare situation, let's say you're in intensive care and you've got like 15 different sensors plugged into you, and they're all measuring you some physiology thing at say 1 kilohertz. So you've now got 15 kilohertz signals coming out of you. And that's just a bunch of data to be processing in real time. You need to be able to tell the nurses and the doctors, like, this dude's about to die unless you stab him with some other drug or whatever. I don't know much about medicine, by the way. As far as I know, those things are supposed to alert people to do different things. And they don't work that well right now. Veracity, a lot of the data that we get is super crappy. We kind of measure whatever we can measure often and hope that is meaningful, but it's often not at all meaningful, and therefore very noisy. Or there's horrible artifacts because, you had this pulse ox thing on your finger and then it fell off. And so it was just measuring nothing. It said to the nurses that you were dead because there was no heartbeat, but that was wrong. So there's a lot of noisy data. That's true for all big data. In biomedical data, there's also this particular issue of domain knowledge, where you have to understand basic things that are going on. For example, if you see no signal from the pulse ox, if you know that it's the pulse ox, then it probably came off your finger, not your head, because even if you're dying, it should slowly. Right? So if you have domain knowledge, it can really help. And if you don't have domain knowledge because you just have a background in statistics or computer science, then it ends up being much better. Okay, so that's why it's hard. There's a couple principles to doing it well. Those of you who have taken my class know these, hopefully. The first one is keep it simple, stupid. I don't know why the word stupid needs to be there. Some people say it, but it could just be keep it simple. Why are we being insulted? So keep it simple. Do the simplest possible thing first. And then there's another concept, principle called look at it, where the basic idea is you should always look at your data in many different ways in detail when possible. So you guys want to see a short video explaining the importance of looking at your data? I don't know if the audio will work.
What, you wiping the windows? Oh, well, look at that. That's nice and shiny.
It's a 78.
Yeah, look really good.
So the
Look
car
really
is the
good.
data. Can you guys hear?
Yeah.
The car is the data. And this guy is a data
Guy in the
scientist.
New York garage every day of its life.
Oh, go figure.
Yeah,
Look
he's
at that.
looking at it.
Oh, look at that.
It's real clean. They can work on it. It's whatever. The things are not packed real.
Right.
There's still a lot of room.
You still do it. Look at that. Look at that. Yeah, look at that. You know, when I see a car like this, first thing I do is I say, would you look at that? You know what I mean? Oh, would you look at that? Would you look
You
at
guys
that?
see that he's looking at the data.
Just look at it. Just look at it. What the heck is that? Oh, look at it. You're running all around the car.
He looks all around the data, right? Not just the
Just
front, the
look
side, the
at
back.
that. Just look at that. Would you just look at it? I mean, just look at it. I mean, just get a look at that.
A cop did that.
A cop, oh, where is he? I'd like to look at him.
All this side information that may not be in your data, but
We're not
may not
required to post warnings here in the jury.
Oh, would you look at that? Listen, I got pulled over in Jersey twice for not wearing a seatbelt in the passenger
Oh,
seat.
yeah.
In the passenger
Oh,
seat.
really?
I got a $40 ticket.
Yeah.
I said, would you look at that? Would you look at it? I was so mad.
Basically, they got to earn their money the old way.
Yeah. Whenever a cop writes you out a ticket, the first thing you do is you take the ticket and you say,
I recommend
look
you
at
watch
that.
the video.
You know what I mean?
Yeah, well, find it.
Test may have questions about that. Okay. So those are the principles. You guys got that one? Principles. Okay, great. So the deal with big data and data science is, as far as I can tell, you need tools to actually do it. And there's lots of ways to divide the world. I happen to have divided it for the purposes of this talk into 7 different kinds of things that you need. Basically, there's 7 things that you always need, or you always need a subset of them. Some of them you literally always need to do any data science, in particular any biomedical data science. And so most of the rest of the lecture, we're just going to go through what each of these 7 things means and why it's hard and interesting in the context of biomedical data science. Okay, so you need to manage data. It's called data science or big data, so there's going to be data. So the way you normally would manage data is, whatever, someone emails you a file, and then it's on your hard drive, and you're done. But if it's really big, then maybe it's too big to fit on your hard drive. So then maybe it's in Dropbox instead, because it's in some remote server. Or if it's really complex, like there's lots of metadata that you care about, you want to search over. Like all of the images you've ever taken, all of your photos, you may want it to be in Google Photos so you can like search for puppy or whatever and get all the puppies in your collection. So with little data, these things you often don't really think about at all. You just store the data on your hard drive somewhere in some file system in some folder. But when it's big data, that doesn't work because you literally can't do it because there's too much data. So you need to do something else. For that reason, people have built databases for the last several decades. There's a bunch of different kinds of databases. There's relational databases, there's something called NoSQL databases, and spatial databases for spatial images. I'm going to talk a little bit more about the spatial databases because that's what I know best, but there's lots of important examples of the other. So in brain sciences, we get lots of data, and in particular, it's often images. And they're images of the brain, and this thing here is a big cube. So one thing we can do is take a mouse or whatever, chop its head off, and then cut its brain out, and then slice it into 80 little slices, and then image each one, and then reconstruct it into a volume. So we get this dense, three-dimensional volume image of the brain. If we stain it in a particular way, then we can see all the little stuff in there, cells and processes and even vesicles and neurotransmitters. You guys know neurons and stuff, right? Okay. So we can see all the bits and neurons. And the deal is that when you're looking at it, you'll often want to look at, say, this part of the brain and the next part, it's like right to the right of it, at about the same time, rather than this part and then this part. So if you're, imagine you're panning or you're zooming around just like you would in Google Maps, but you're navigating the brain, you want to be able to move kind of locally easily. And so it turns out that disk access takes time. Every time you access data on the disk, it takes time. And so what you want to do is you want to organize your data in such a way that data that's local, meaning this pixel is close to this pixel in brain space, should also be stored locally on disk such that you can just kind of keep going to the next entry in memory rather than finding another place in memory and loading up more data. And so that's what a spatial database does. We happen to have built one. It's the biggest one in the world for brain data. We've got several hundred terabytes of data. Voxel is a three-dimensional analog of a pixel. It's a voxel element rather than a picture element. It's got hundreds of public and private data sets and lots of collaborators and lots of 3D data, but also 4D, 5D. So that is a kind of database. There's lots of them. By the way, all these things I'll show you, there's links down here. You can go check out the documentation for the databases. Okay, so that is a way of managing big data for this particular context. If you have different contexts, different queries, you would do it differently. Is that clear? All right. So you always need to manage data, and then you also always need to visualize your data, because you remember the principles, you gotta look at it. So usually if you have little data, it's not a big problem, right? Someone sends you some data, you can look at it and say, draw a plot in R or MATLAB or Python, or if it's an image, you just load it up in Preview or MS Paint. Is that still a thing? How do you open? Someone told me last year. How do you open images in Windows now? Is there a thing called MS Paint? That's what you do? All right. Anyway, or ImageJ or whatever. There's lots of local clients that let you open images. But now let's say your image is 100 terabytes, which we have multiple images that are 100 terabytes. You can't just open them MSP or ImageJ, you have to do something else. And so an option is to essentially have a graphics library that is written in some language. So I gave a couple of examples here, WebGL and D3.js. They're basically web libraries. And the deal is the data are stored somewhere, and these libraries interact with the database and pull the data that it needs and dynamically generates the image for you. So in the case of, say, Google Maps or our stuff, the data is stored on disk in some compressed file format, not in an image. But this library will pull the bits that it needs, and it will render, say, a JPEG or a PNG image for you to look at. Okay, and that's how you can visualize big data. And it doesn't matter what the format of the data are or how you want to image it. It all basically works in that principle where you store it somehow, and then you use a library to dynamically render the images. Does that make sense? You're not actually storing images off it, or whatever format you want to look at it. One of the nice things about the database is you can set it up such that you can call and get different file formats that you want. So if we're storing data that are images, you can set it up so you can get JPEGs that are compressed, or PNGs that are uncompressed, or bitmaps, or lots of other file formats, different sizes. Okay. So we have something we use called NeuroGlancer. This is just a screenshot from an image. The black and white stuff is electron microscopy of a small part of the mouse visual cortex. And so you can see a bunch of stuff. This thing, these solid things here, this is where we manually label that there are synapses, which is where neurons are connected to each other. The kind of blurry green and purple things are markers of different proteins. I think the green is synapsin, which is a protein in basically every synapse, and the purple is another one. I forget which one, maybe GAD or something. It's some synapses, not all of them. Anyway, this tool we have, which really is just like Google Maps, but lets you do stuff in 3D and lets us overlay. If you know in Google Maps, you can overlay, say, the street names or topography or stuff like that. We did the same thing where you can overlay, if you have lots of different channels, you can overlay them on top of each other, including manual labels and stuff like that. That make sense? Then there's wrangling. So data wrangling is kind of a more recent term. It's a little bit vague what people mean. So I define it as you take your data, it starts in some format, say an image, and then the output of it is another image. Right? So for example, things like if there's missing data and you impute the missing values, that's data wrangling. Or if you convolve, do you guys know what convolutions are? You took signals and systems, right? Okay, so if you convolve it, involve a time series, you get out a time series. If you find that there's an outlier, it's because when you were doing your imaging or experiment, someone kicked the table, and so the thing went off the charts. Detect that there's an outlier and take them out, but you still have the true signal. That's also data wrangling. And people have been doing data wrangling forever. If it's really little data, you often just do it in Excel, right? You've got a data table and you find someone wrote -8 somewhere, and you know the data is positive, so you can go. or you do some scripting. When the data are really large, like 100 terabytes, you can't do it that way anymore. What you need is these distributed pipelines, and you run them in things called scientific workflow management tools. And there's a bunch of them. Galaxy is one that's actually developed by this dude here, kind of awesome guy, in the biology department. It's basically the best bioinformatics scientific workflow management tool, so it's specific to optimized for scientific workflow for bioinformatics. NiPipe is one for neuroimaging. Luigi is one that Spotify developed. You guys know Spotify? Yeah, like music service. So they have to do this too, right? They've got, I forgot, maybe 100 million songs now. And so they've got to route different songs to different people when they want them. And so there's a bunch of complicated pipelining. And so they developed something called Luigi. I assume it was named after the Super Mario character. Also named something Luigi. But it could be something else. Point is, you need something like this, or some terrible hacky thing with bash scripts that isn't. Okay, is that clear? The data range is something. Basically, always need to do it whenever you get raw data. The first thing that happens is you rank it. Okay. So here's another example from our lab. This is, we have this data set, it's called a clarity range. So clarity stands for clear something brain. I don't know what it stands for. Obviously, it's just because it's clear. So you take this brain, the way it works is you take a brain out of a mouse, say, and then you put them in a bucket, the brain. It's basically a vat of chemicals, and then it sits there for a month. And then you take it out, and the whole thing is clear. So you remove all the lipids that are opaque, and you replace them with basically plastics in this process. And then what you can do is if you've done things prior to that to make certain cells fluorescent, then you can do optical imaging, and you can see all the fluorescent ones throughout the entire depth of the brain. So now you can get 3D volumes of a few neurons that are sparsely labeled, and you can see the whole thing. You never have to physically section the thing, which takes a bunch of time and money, and then you have to do this reconstruction, which are really onerous. And so there's a bunch of ways to do it. One of them was developed actually by a PhD student at Hopkins, Zhu Hao. It's called iDisco. It's nice too. But anyway, what you want after you do that is to figure out things like, okay, all these neurons that I can see, what parts of the brain are they in, and if we're visualizing, say, their axons and dendrites, which you guys know what those things are, we want to know where they go. So let's say we figured out that we fluoresce all of the dopamine neurons that come from a particular region of the brain. We want to see where they all go. Because no one really knows, because usually you have to slice the brain, and then you just look at where it goes within a slice. So what do we do? Well, here's what the sample image looks like at first, and you can see it's crappy. Right? This whole thing is super bright, and then as you get further away from the middle, it gets dark. So the first thing we do, which is brilliantly named pre-process, is we pre-process the data, and we kind of histogram normalize. So now, basically, we correct some of that stuff. It's actually pretty hard to tell on this crappy projector from like 90%. But if you all gather around over here, see?
Oh, yeah.
Yeah.
Well, you guys have links to the slides, right? So you can. It looks nice, yeah. Okay, then the next thing we do is called affine registration. So there's an atlas that the Allen Institute built for like $30 million. They took something like 100 brains and they averaged them all together. And they built this atlas, and that's their third thing. And so now we want to register our brain to this atlas because someone went through painstakingly and drew little circles in the atlas to say, oh, this region is visual cortex, and this is hippocampus, and this is unlabeled, which is, turned out a lot of the brain is unlabeled. So the atlas is problematic. Nonetheless, it's the best one that exists. And so what we do is we take this brain that we have, and we do an affine registration to it, which means we just shift it and rotate it and rescale it, and to get it basically lined up with this, with the Allen atlas. Then what we do is this nonlinear thing called large deformation diffeomorphic metric mapping, which is almost as brilliantly named as preprocess. And what it does is it does a nonlinear registration to squish the brain, our brain, to be much closer to the shape of the atlas. Then once we have that, it's really easy to just map from the atlas, which has all these circles drawn onto it, where they are on our brain, and then we can see, oh, all these cells that we can see, which regions they're in. Okay? Is that clear? This whole thing is a big mess because this raw data starts out as like 20 to 40 gigabytes. So you can't just do the whole thing on your computers because, like, you know, that laptop over there probably has 8 gigs of RAM, so you can't load it. You have to do something more complicated. And so we have this kind of distributed pipeline that does parts of it at different times, and it's very fancy. And this is the output. It looks happy on this projector, but it is tricky. Okay. Then you summarize. Now that you've done all the pre-processing and everything. So summaries of your data include things like the average height of the sample was 8 feet. And that's maybe if we were sampling groups. How tall is Groot? Does anyone know? Remember? From Guardians of the Galaxy? I guess, I don't know if all, I'm guessing all of those guys have the same group, but I think the average group would probably be 8 feet. If you sampled a whole bunch of groups, maybe the average would be 8 feet. That's a summary of groups. There's also another kind of summary which happens in image processing, which is, in this box is an elephant, which is a summary, because there's a whole bunch of pixels there, and now you said it's just an elephant. Or in speech and language processing, you may say, this is a noun and this is a verb. Right? It's another kind of summary. So those two different kinds of summaries, there's two fundamentally different kinds of summaries. One of them where you maintain the representation. You measured heights of groups. What's a species of group? I forgot what it's called. Tree. I don't think a tree is a species. Also, I'm pretty sure he wasn't a tree. I think he's like a magic something.
Flora colossi.
Say it again?
Flora colossi.
Really?
Yeah.
So the flora, colossus, like he's a colossus flora? That's cool. So, anyway, if you measure the height of all the floras colossi around, and then those are just numbers, right? There'll be some non-negative number. The mean is a summary, it's the same, it's in the same space. You measure numbers and then you output them. But if you see that, if your summary is that there's an image and the tree is in the upper left quadrant, you've converted it into a different space or a different representation. It started out as a bunch of pixels and they had intensity values from 0 to 255, say. And now you say this whole thing is not in that space anymore. Now it's some semantic label like tree. It's another way of summarizing it. It's a fundamentally different thing. It's often harder, but it's also simple. Okay, so here's a version of us summarizing essentially that same brain. It's not quite the same brain, but what we did is we ran some machine vision algorithms and we detected the location of every cell body in the brain. And so all these yellow dots are those locations. But so the brain started as, this one was a 20 gigabyte 3D volume, and now we've summarized it as a collection of points in 3D. So there's like 1,000 of them, so it's 3 times 1,000, so 3,000 numbers, and it started out as 20 gigabytes. Right, so that's a pretty good summary. It's obviously not the same space. The first one was an image. This is a list of points in 3D. Nonetheless, it's a summary, and it's a nice one because it has the semantic content that we want to be operating on. We want to study, say, the spatial distribution of cells in this range. Right? We don't really want to study the image per se. The image lets us estimate these other summaries that we can then use in the analysis. Okay? Okay. So, common is the dude, he works in the business lab at Hopkins actually. essentially building smart bombs, trying to figure out how to control rockets to blow up more or less brown people. I think it was Koreans at the time. Could have been Vietnamese. Anyway, some brown people that are trying to blow up. And so they did the whole common filter thing where they can figure out how to control these things. You can have moral issues with that, but it founded the theory of control theory. Then there's reinforcement learning, which is getting a lot of hype recently because DeepMind plays video games better than people now. I don't know why that's interesting, but it makes a lot of hype, and you can use reinforcement learning to train algorithms to do better than people. It turns out the way they do it is because they can simulate games. You can simulate them at like really high rates, faster than people can play. And so basically the algorithms just play like 100 million times more than any human could ever play. All right. If I played 100 million times more, I'd do better too. But it also turns out they burn down like a rainforest every time. It's like a whole order of magnitude more computing than anybody else has ever used to try to beat. Okay, design of experiments. This is actually one of the most fundamental things in statistics. Figuring out how to design an experiment so that you can make a general statement. There's randomized controlled trials and case-controlled studies, and there's active learning where you're dynamically choosing stuff. And there's a couple of libraries people are recently developing. Google's dopamine. It's named partially the reinforcement learning is actually part like kind of behavioral theory from people studying humans, and then machine learning, and so dopamine is a neurotransmitter in your brain that seems to be implicated in learning how to do stuff, because when you get it, you like getting it, and so you'll do the thing over and over again. So if you put a mouse in a room where they can have heroin or sugar, they end up taking heroin because it kind of gives them more dopamine than stuff. So that's what Google is going for. Sorry for that, but they'll make a lot of money when they do that. And then OpenAI Gym. OpenAI is this like non-profit artificial intelligence institute. They built this gym environment to let video games try to beat, well, let algorithms try to beat people in video games, essentially. Okay, I haven't ever worked on that, so I don't have anything particularly interesting to say about that. But do have something to say about the next vistas. This is, I find, really cool. There's a paper in PNAS several years ago by Don Keene, who's here at Hopkins. He's one of the most important statisticians in the 20th century. He came up with these tests. He didn't call them visual Turing tests, but they basically are. Basically, you would see a couple examples from this or this. And you have to figure out what is different between this one and this one. So do you guys, you've now seen one training example. Do you know what the difference between this one and this one is? What do you guys, what does that do? What? Yeah.
Orientation and also two shapes aren't the same, like in the first topic.
In the first, these two shapes are the same, and this one, they're not. Okay, so that's an example of something called same versus different. It turns out some of you can do that with one training example. And the best machine learning algorithm in the world can do that with never. You give them an infinite amount of training samples and they just don't learn. This concept of sameness is this relational concept where you have to first identify the objects and then understand the relationship between objects. No things do that now. And so this plot over here, this is a paper published last year. Out of these 23 problems, there were like 10 or so were same versus different kinds of problems, and the other 13 were not. And what you can see is for the same versus different, this is after something like 100,000 or 100 million training samples. There's no algorithm that did particularly better than chance. And for all of these, humans get it right after every human. Even those of you who didn't say it after one, you will get it right after like 10. So machine learning and deep learning totally sucks at this. And partially because it's this complicated thing where you have to convert the data from one representation into the other. Like I was saying before, you got to do this object recognition thing. Then there's this whole other thing about understanding the relationship between the objects that really no one has anything to understand particularly well. So machines suck at that now, but they're working on it, and they'll probably be okay at it soon. But that's kind of, I think, one of the big next vistas for biomedical data science. And these are references for, we wrote papers and all these things I showed you, and so you can read them if you want. And this is a bunch of people who worked on that stuff. And that's a cartoon image of my son. Turns out, in iPhones you can take a picture and then dynamically make it look like a cartoon. And so I did that the other day. He was sleeping. That's my son as a cartoon. It's appropriate because you can see here, his name is Lion. It's written there. He was actually named after Lion-O, who was one of the Thundercats. That's maybe too young to watch Thundercats. It was a great Saturday morning cartoon growing up. So it makes sense to have images of my son. Okay, that's all. Thank you.